Very much. I'm Al Smith. I'm the chairperson of the Department of Kinesiology here at Michigan State. And on behalf of the Department of Kinesiology and the African Studies Center, I'm honored and extremely pleased to welcome you to the inaugural Deborah L. Felts Lecture on Sport, Exercise, and Human Movement Science in Africa. This event offers a wonderful opportunity to build partnerships on campus and with African colleagues in advancing scholarship in kinesiology, as well as promoting global health and well-being. We're grateful for the generous gift from Dr. Felts that made this event possible, and in a minute I'll share some background on that. But before I do that, I want to thank all of you for coming today. I want to thank the African Studies and Kinesiology staff also, who helped with various event logistics. And then finally, I'd like to thank our Felts Lecture Selection Committee. That would be Dr. Jamie Monson, Director of the African Studies Center. You'll hear from Dr. Monson momentarily. Dr. May Lee, Assistant Professor of Kinesiology. Dr. Leps Malete, you'll also hear from him. He'll introduce our speaker today. He's Associate Professor of Kinesiology. And Dr. Tayo Moss, a recent graduate of our Kinesiology doctoral program. So thank you, and I appreciate, again, all of you being here today. The endowed lectureship was established by Dr. Deborah Felts, University Distinguished Professor and Chairperson Emerita, who worked with the Department of Kinesiology from 1980 to 2017. And 23 of those years she served as the department chairperson. So she had a huge impact on our department. And having conducted research on youth sports in Africa and mentored graduate students from around the world, she wished to institute this endowed lecture series to facilitate collaboration and dissemination of sport, exercise, and movement science research, as well as promote global understanding and the growth of this research area across the African continent. She seeks to support the formation of mutually valuable collaborative partnerships through this endowment. She is here today. If you don't mind, would you just stand up real briefly so we can acknowledge you? We are grateful for your vision and for enriching our community in this way, so thanks so much, Deb. At this time, I'd like to introduce Jamie Monson, who is Director of the African Studies Center here at MSU. Her group is partnering with us in organizing this new lecture series, and I know she wants to say a few words as well about this event.
Thank you so much. Thank you and welcome, everyone. So I was looking back through my notes, and it was December 10th, 2016. I got an email from our new faculty member, who's really an old member of our community, Leps Malete, and he asked me if I could have a meeting with Deborah Feltz, who I'd never met before, to discuss an initiative she had in mind to bring African researchers and experts on sports and physical activity to MSU. So I got to meet Deborah Feltz later that month, and I learned that on her retirement, on the occasion of her retirement from MSU, she was planning to make this generous gift in the form of an endowment to support an annual lecture. So why was a sports and kinesiology professor in my office in African Studies talking about a lectureship? It was because she wanted to use this gift to continue her lifetime work of building bridges among MSU students, faculty, and researchers and African partners. In other words, Deb Feltz is one of those who fosters our partnerships between MSU and African institutions, and this is a legacy of partnership that she will be continuing to leave with us after her retirement. So thank you so much for that vision. Here at MSU, partnership is really the heart of our engagement with Africa, and this is a wonderful example of partnership in action, as we have MSU, African universities, faculty, students, and above all, mentorship among our different institutional relationships. But not only that, this is the year of Global Africa at MSU. So what a fitting time to have the inaugural Deborah Feltz lecture than during our Year of Global Africa. We are in fact celebrating with the Year of Global Africa our legacy of connections. So I'm especially honored to welcome all of you here today for the first Deborah Feltz inaugural lecture. Our speaker is Alison September from the University of Cape Town, and I'm going to be leaving her introduction to Leps Malete, who will introduce her in a minute. But let me, before I finish, extend another invitation for those of you who'd like to join us on Thursday at noon downstairs on the second floor. We'll be having at our normal Eye on Africa time a Q&A with Dr. September, talking about broader issues about higher education in Africa, especially Southern Africa, related to research, institutional transformation, diversity, and inclusion. So we'll be broadening our conversation. Everyone is welcome to join us on Thursday at noon. So let me thank you all once again. Thank you to Dr. September for coming all the way from Cape Town, to Deborah Feltz, for all of you for coming today, and I'll hand over then to Dr. Malete.
Thank you very much, Jamie. This is really an exciting day for all of us because we are also celebrating the power of partnerships, networks, what networks could do in our lives. I have the singular pleasure and privilege of introducing this afternoon our visiting professor and presenter for the day, our speaker for the day, Dr. Alison September. Dr. September is actually associate professor in the Division of Exercise Science and Sports Medicine at University of Cape Town. This division is actually in the Department of Human Biology, the Faculty of Health Sciences. She has a PhD from University of Cape Town in genetics, and her thesis was actually on molecular investigation of Stargardt disease in South Africa. She's actually done some very fabulous work at this institute that I know very well, and I've heard a lot of good things over the years. One of the things that she's specifically interested in is the identification of genetic risk factors underlying soft tissue, such as Achilles tendon injuries and anterior cruciate ligament ruptures. She's specifically interested in how the role of genes could actually help in encoding, role of genes in coding and proteins, how they help with the remodeling of tendons and ligaments. What she's also interested in today in sharing with us is the work that she's doing and how it is actually also helping with the young people, the youth in Africa. In particular, she's interested in mentoring young people, young women who are coming from less advantaged backgrounds. So this is actually very powerful work that she's doing in terms of transformation, not only through her research, but also mentorship of other individuals within South Africa. and I believe that her work has global reach. So we are very excited to have you here, Alison, and I believe that you are ready to give us a presentation, and you have a very good audience today. Thank you very much.
Thank you.
Yeah.
So I'm very humbled and honored to have been invited as the inaugural lecturer for the Professor Fal's inaugural series, and I'm looking forward to engaging with you, and I've designed my talk so that we can build partnerships with the University of Cape Town and MSU. In my presentation today, I'm not speaking to you about my work on transformation, but on Thursday I will share with you on my many activities both within the University of Cape Town, nationally and regionally, as well as internationally, what I'm doing on transformation and the youth and women in particular. So lessons from the Genomic Code. So the few slides is just to show you about my hometown. On the top, here's my pointer. Over here, this is our famous Table Mountain, and as this picture looks, that's how it is. I'm a little bit homesick. And this is the mountain from the Bloberg beach with the skylights. This is where I work. This is the Sports Science Institute, and if you've been to Newlands, the Newlands Rugby Stadium is right next to the Sports Science Institute. And this is the main campus of the University of Cape Town, which is on the foothills of Table Mountain. So during lunch, you can always go for a run up the mountain. And often our professors at work, the crazy bunch, before work starts at 8, they would have gone up to the Lion's Head or somewhere and start work at 8 o'clock. I'm not part of that gang yet. So my talk layout, I'll give you some introduction. Speak about tendons and ligaments, the basic structure. Can you see this pointer? Some of the experimental work we've done recently, which has been published in 2016, 2017, and 2018. Some of the functional work that we've done to support the statistical analysis that we've conducted, and some of the risk models, as well as our new data on whole exome sequencing that's not been published but is in review at the moment. And I'm going to give you the new approach we've tested and give you an example of one of the genes we've applied it to. So, as you know, I don't have to give you much detail, but the incidence of lifestyle-related diseases are ever increasing in the developing world and the underdeveloped world, in the developed and the developing world. And as clinicians and as scientists, we're trying to understand what are the driving factors causing these rise in the medical burden or this disease burden. And as sports scientists and clinicians, we see a spectrum of these diseases, ranging from the elite athlete, the recreational athlete, the sedentary individual, as well as the recovering patient. So we go from two extremes, the performance extreme as well as the diseases. And we're trying to understand right down to the cellular and the molecular level, what are the contributing factors predisposing these individuals to these diseases, what are the predisposing factors protecting some of these individuals from these diseases. And to achieve this, it's why I'm here today, is to follow a multidisciplinary approach, a collaborative approach, where we start from the gene within the cell, see how those genes and proteins work within the cell and how that comes together to produce the function or dictate the function at the tissue and organ level, resulting in your clinical presentation, which is either your phenotype injury or your non-communicable disease. And so we need several specialists to communicate, to interact, to collaborate, to understand the disease mechanisms before we can prescribe intervention therapies or strategies. And so as we move towards improving the effectiveness of healthcare, we need to collaborate. We need to talk. Basic scientists have to collaborate with clinicians and surgeons before we can predict and apply a risk management strategy. But the first step to understand how we can go from a healthy tissue to a diseased tissue is to know the knowledge out there, how much of the knowledge out there is actually correct and what is actually wrong. And so that we need to acknowledge that some of the knowledge out there is not it's not correct. And we need to improve that knowledge, the correct knowledge. And then understanding that collectively, can we start to accurately diagnose and improve the clinical management of these diseases, so that we can accurately increase our ability to predict disease, to predict susceptibility, and to manage their conditions. And the trend in medicine is to conduct precision medicine. What is precision medicine? It is to treat the individual based on the individual's clinical characteristic, the individual's needs and preferences and lifestyle. And for that, we need to understand our knowledge barometer. What is correct? What do we not understand yet? And very important, we need to understand the limitation in our knowledge. So what is wrong? So my area of interest is connective tissues. And connective tissues are commonly injured in different activities. They can be sporting activities, organized or physical activities, and even there's an increase of connective tissue injuries at your workplace, your occupational activities. And broadly, they can be classified as either acute onset conditions or chronic overuse conditions. An example of an acute onset injury that we study is acute ruptures of the anterior cruciate ligament of the knee, as well as spontaneous ruptures of the Achilles tendon. The overuse injuries that we study in our laboratory is the chronic overuse Achilles tendinopathy, and specifically the mid portion of the Achilles tendinopathy. What do we know about these injuries? Well, we often say in research that they are multifactorial conditions, and we say that when we don't understand much of it, and that there are many, many factors which interact together with each other to produce the disease and to increase an individual's susceptibility to the disease. What do we know? That there's much clinical heterogeneity in the pathology of these connective tissue injuries related to pain. There's much variation between pain, that the amount of pain doesn't correlate to the pathology we see within the tissue, as well as the clinical presentation, as well as the irritability of the specific connective tissue. And so there's much heterogeneity in the capacity of the tissue to withstand load, how the tissue recovers from load, and how people respond to specific treatment. So if the Nobel Prize is out there, if we can understand and unpack what are the factors, biological factors, clinical factors that contribute to the optimal functioning of a connective tissue. What do we know? That tendons and ligaments are similar in structure, although they are anatomically distinct. Tendons and ligaments are basic collagenous structure. The major component of tendons and ligaments is the type 1 collagen. 2/3 of both these tissues are comprised of water, and tendons are specifically packed in these hierarchical structures made up of type 1 collagen fibrils, and they aggregate together to form larger structural units. And you can see in this pictogram, you can see the beautiful, I'm going to use my this is much better. You can see the beautiful, tightly packed parallel bundles. And if you look at a disease histopathological examination of these tissues, you'll see that beautiful parallel structure is destroyed or distorted in certain areas of the tendon. And so as biologists, we're trying to identify what are the factors causing that destruction, and why are some people healing and others are not healing. So then, besides type 1 collagen, there are many other minor collagens present which make up your collagen fibril. Examples are they can be broadly classified as the fibril forming collagen types, the fibril associated collagens with interrupted triple helices and beaded filaments, etc., etc. Our lab has studied type 1 genes encoding type 1 collagen, type 5 collagen, as well as type 12 and I think 19, I can't remember, and 27 collagen. There's also minor components include glycoproteins such as uteinasin C, comp, elastin, fibronectin, and many of these proteins either intercalate directly with the collagen fibril, or they associate on the surface of the collagen fibril, or they allow for communication between fibrils or between fibers. And so even though they are the minor composition within the tendon or the ligament, they play a very significant role in maintaining that fibril structure and assisting with the communication between a load, response to load, recovery from load. Proteoglycans and glycosaminoglycans also make up a minor component of your connective tissue and play a very important role in contributing to the structural integrity of your tissues, but also contributing to the cell signaling of these tissues. And so I have a particular, my graduate students will always laugh at me when I say proteoglycans is my favorite genes to work with, because they are absolutely phenomenal. When the one is down-regulated, you always find another proteoglycan is able to compensate for that function. And so if we can understand what are the key proteoglycans, I think we can probably suggest improved treatment for these injuries. And so some of the proteoglycans that you can find is your small leucine-rich proteoglycans, such as your decadent bioglycan, fibromodulin, and lumican. You also get larger aggregating proteoglycans such as agrican and versican. And so this is your large aggregating proteoglycans. It almost has this bottle brush effect, and I'll show you another diagram of how they facilitate communication between your fibrils. Okay, so some of them, and so your large component of your tendons and ligaments is also your ground substance. And so you would, when there's an injury, there's inflammation, and there's an increase in your proteoglycan and GAG contents, which attract water into between your fibrils and assist with the reaction to the load. Okay. So if you compare connective tissues such as tendons and ligaments to muscles, you'll see a different picture. In muscles, your cell is your major component of your tissue, but with tendons and ligaments, your extracellular matrix is your major component of your tissue. And so your cells form a very small component, however they're quite powerful within your tendons and ligaments, that they are able to sense load, they're able to send a message to the nucleus to produce all the components of your extracellular matrix, thereby maintaining the integrity and structure of your connective tissue. And so even though they are small, and small in the sense that they are minor, they play a major role in what we say regulating the matrix remodeling. And so we try to understand, how does the connective tissue go from a normal connective tissue responding to load to a diseased state. And sometimes once you've gone through to the disease state, no amount of therapy can rehabilitate you. And so with chronic Achilles tendinopathy, there is a large proportion of these patients who remain symptomatic throughout their lifetime. And if you speak to a physiotherapist or a biokineticist, it is really sometimes difficult to treat some of these patients, and we're trying to understand what are the driving factors to move them into the disease state, and what are the factors that allow some individuals to react appropriately to a load and to recover from that load. And so the tendons and these cells are able to respond to several stimuli, send a signal to within the DNA to produce the components within the extracellular matrix, which is required to maintain the tissue integrity. And some of these signals are produced to produce more collagens, to produce more components, to maintain the healthy fibril structure, which is type 5 collagen, type 1 collagen, your proteoglycans, your glycoproteins. And there are also other enzymes within the structure which main duty is to regulate those components. So at some point in time, we don't want more collagens being produced, and there are these matrix metalloproteases abbreviated to MMPs, that are specifically targeting specific different types of structural components, and they get degraded. And so there's a balance or a homeostatic regulation between the synthesis and degradation of the extracellular matrix components. And a lot of research is trying to understand what is that homeostatic regulation, what is the fine-tuning of maintaining that homeostasis of the extracellular matrix. And not surprisingly, there are also proteins within the structures which regulate your matrix metalloproteases, and those are called TIMs, tissue inhibitors of matrix metalloproteases. An easy name to remember. And so cytokines, so chronic Achilles tendinopathy is not regarded as an inflammatory condition. However, there are still inflammatory processes that are happening, which are called cell signaling, which is critical to assist with the cell or the tissue to respond to load, to activate a whole biological pathway so that you can respond to the load, that if there's any damage to your fibril, that can be corrected. And so we need to understand what is that first line of defense, and I believe it's our interleukins together with our proteoglycans. And you have many other factors. And so those tissues which are able to have, so we try to understand what determines capacity to withstand load. You have to have an appropriate response. Timing to response is also very important to facilitate repair, healing, adaptation. And then what are the factors to determine pathological response? So you get dysregulated cellular interactions, dysregulated cell signaling. So with the normal response, you get areas of synthesis and areas of degradation, and somehow that regulation is disturbed, and you find that you have some areas where you get decreasing cellularity, which is facilitated by a programmed cell death called apoptosis. And in other areas of the tendon or ligament, you get areas of increased cellularity, which is good because you want more cells to produce more components to fix the structure within your tendons, and that is called hypercellularity. And so if you look at a diseased tendon, you'll find different areas of increased cellularity, decreased cellularity, increase of damage, increase in repair. And so when you go to a pathological state, that balance is disturbed. And so the continuum model has been proposed by Cook and Purdon from Australia, where they say, so when you look at the inactive population, you also see that there's an increase in chronic Achilles tendinopathy in these patients. And also with diabetic patients, there's an increase of tendinopathy. And so a healthy tendon, the saying is that a healthy tendon requires a healthy load. And so healthy tendon exposed to an individual will have its own, because every connective tissue of an individual, well, if you compare individuals, their connective tissue will have a specific structure, and so they will respond very differently to load, correct? And so you need to understand what is the appropriate load for the individual. The tendons will respond to that load. You get cells sense this load, they produce more cells so that they can repair if there's any damage and get activation of the cells. And once that repair is maintained, homeostasis is regulated, you have a normal tendon and you remove the load. However, in certain cases where you persist with the load, you increase the frequency, it results in tendon disrepair. So the cells are unable to cope with the amount of damage anymore, and it causes damage to your tendon. And that is when you get a degenerative tendon, and you cannot go with any length of rehabilitation, you cannot go back to a healthy state, and your tendon is actually compromised. So that is the continuum hypothesis model. And so we think that these cells, even though they are a minor component of this connective tissue, that communication, so even cell-cell communication is pivotal in understanding and regulating this matrix remodeling of our tendon and ligament structure. And so here's a cartoon that I've illustrated to you. This is your collagen fibril. These are two collagen fibrils you have proteoglycans, small leucine-rich proteoglycans, which are able to either attach to the collagen fibril, thereby regulating the process of fibrillogenesis and the structure of the fibril. You have these large bottle brush, those who are moms or dads who are used to cleaning bottles in the good old days when there's still bottle brushes, that there's this bottle brushes effect that maintain cross-talk between the fibrils. And so cross-talk is important to allow communication between cells as well as communication between fibrils to maintain the tissue integrity of these connective tissues. And part of our genetics work, we've targeted these different components of the tissue to try and understand how does genetic susceptibility contribute to the disease phenotype. So we know that there's clinical heterogeneity and how individual connective tissue responds to load, the amount of force and the frequency of the force. And so we're trying to understand what are the factors at the molecular level which contributes to the capacity of tendons or ligaments to withstand load and the frequency of load. Research has shown that these conditions are multifactorial, that there are many, many different risk factors. We use the term intrinsic risk factors, factors within the body, and extrinsic factors, those are factors that you expose the body to, and that genetics is one of these small components contributing to the susceptibility. So genetic compared to a monogenetic disease does not, it's not a cause disease relationship, but it contributes to your susceptibility and how you present as a phenotype. So there are many, many factors contributing it, and we're trying to still understand what are these factors, how does each of these factors weight in the disease model, and how do they all fit together. So we have a long way of understanding that. And so the genetic sequence is known for quite some time now, and we know that we are 99.9% identical at the sequence level. And so biologists try and exploit that 0.001% between us to try and map what we see at the DNA level, which we refer to as the genotype. So the variability at the sequence level is called the genotype, to what we see at the biological variation, either the disease response, which we term in genetics as your phenotype, so your clinical phenotype. What is a genetic variant? A genetic variant, if you have a sequence of DNA that if for example this is a C and you have a substitution for another base at that point, this is called as a single nucleotide substitution, or an SNP. So a sequence variation is just that between you and me at one position in our stretch of DNA, there's an alternate sequence at a single position. And those variations, single nucleotide polymorphisms, occur on average about 100 bases, so they are very frequently distributed throughout the genome. And they do not cause the disease, it's not a cause-effect relationship, but they may affect your how you present with a phenotype or affect your predisposition. So if you have genetic conditions, for example, Duchenne muscular dystrophy or Huntington disease or retinitis pigmentosa, or certain cancers, if you inherit the disease-causing mutation, chances are that you're going to present, the phenotype is based on the prevalence, not prevalence, the chances are that you're going to get the disease could be 99.99%. So if you get the mutation, depending on the age of onset of the condition, you may have it. So there are very few genetic conditions where it skips generations. Okay, so we try to correlate what we see at a sequence variation to how it may influence the RNA and thereby influence how much protein is made, not necessarily the function of the protein, but how much protein is being made. So either more protein is being made with a specific sequence variant, or less protein is made if you have a specific sequence variant. And thereby, if you think about these proteins are made within cells, the cells will then act differently to the response to load, which will then affect the tissue integrity and therefore affect the disease progression or the disease phenotype. And so that's how we modulate what we see at the sequence level to what we predict to see at the clinical phenotype, the biological variation. And in the literature, they mainly conducted case control study designs. So we have a population of affected individuals, and we have matched cases where they have a similar exposure, yet they are older, but they do not have the disease phenotype. And to date, there's about 80 different polymorphisms have been identified to reside within at least 30 genes to be associated with either an increase or decreased susceptibility to Achilles tendinopathy or ruptures of the anterior cruciate ligament. And so there are several structural components have been implicated, and these are the gene names. Several proteoglycans, regulators of the matrix, your matrix proteases, those involved in apoptosis. Apoptosis, which we spoke about earlier, is a programmed cell death, which is one of the disease status. If you look at the histopathological examination of these tissues, you'll see that there's apoptosis, that there's cell death within specific areas of the tissue. And several of the genes encoding cytokines and signaling factors have also been implicated. However, if we know that the genome consists of over 3.2 billion sequences, that we're only implicating a very small proportion, and it's not only going to be those 80 loci, so there's a huge amount of data that's still missing. So I'll speak at the end how we're trying to identify that missing heritability, missing susceptibility genes. Some of the pathways that we've investigated is the angiogenesis signaling pathway. Angiogenesis referring to new blood vessel formation. Both tendons and ligaments have a poor vascular system, and so why do you think, you may ask, why did you then study the angiogenesis cell signaling pathway? Well, because angiogenesis plays a vital role in the repair mechanism of these tissues and facilitates the response to load. And so there are several genes functioning in this new blood vessel formation pathway, and the two that we've studied was the vascular endothelial growth factor, with its receptor being the kinase domain receptor. And some data that we've looked at two functional polymorphisms within the gene called the vascular endothelial growth factor A gene. And over here we've looked at an ACL cohort. We have the controls in the black bars. The white bars are our ACL cases. And we've stratified our samples into two mechanisms of injury, the contact mechanism of injury and the non-contact mechanism of injury. Non-contact, our hypothesis would be have a higher intrinsic component, so the genetic component should be higher compared to your contact mechanism of injury. So the ACL group contains both contact and non-contact, contains the whole affected cohort, and the non-group, it's only the non-contact injuries. And so this is the specific polymorphism. The CCGA and the AA, CA refers to the genotype variation, and the frequencies on the Y bar. So we found that individuals with a CC genotype at this specific polymorphism, just like we all have back home we call it an ID number, where the government will identify you with, that's your ID number. So on the databases, on the public domains, every single polymorphism also has a unique ID number. So if you search on any of the public domains, you can use that unique identifier and it will be unique to that particular polymorphism. So that particular polymorphism, the CC genotype, we found to be associated with an increased risk of ACL ruptures. The GA seems to be associated with a decreased risk of susceptibility to ACL ruptures. And if you look at each gene will have a unique structure, and the sequence is out there, so we can map exactly where each of these polymorphisms are aligned to. There's functional data for these three polymorphisms that we've looked at, and they reside within this 5' promoter region of the gene. And why is it important for us to know where it is? Because if you want to map a region to explore functionally, if you find that particular region of the gene is associated with a susceptibility, maybe an increased susceptibility or decreased susceptibility, we actually want to, that's just a statistical association, but you actually want to see, well, what is the functional biological effect of that particular region. So if you know the functional effect, it will strengthen or support your hypothesis of the association. So it's not enough just to have an association. You need to show proof of concept of function, as well as to repeat it in different populations, because you don't only want to be treating a certain small subset in South Africa, but you want to understand the biological significance for all of us. And so you want to repeat your genetic association studies in different populations and in large numbers. And so I showed you where we looked at individual SNPs, so one over here, there and there. But it's more informative to look at all three in combination, and that's what we call the haplotype analysis. So a haplotype, when you see the word, is really a combination of individual SNPs. Haplotypes will tell you, it's like a paint. So you paint the chromosome, you'll say, well, that stretch of the chromosome is unique and it's associated with disease susceptibility. So it's way more informative than individual polymorphisms. So we used haplotype analysis, and we see that actually if you have an AAG here, you have more likely to have a reduced susceptibility to Achilles tendinopathy, and a CGC over here in females. So we've also stratified our data set according to sex, because females are at an increased susceptibility to ACR ruptures. And I think for Achilles tendinopathy, it's males, although the jury's out. There's been conflicting evidence to suggest if it's males or females, so I'm not entirely sure as yet. But definitely with ACR ruptures, if you're a female, you're at an increased susceptibility, although the prevalence is lower in females compared to males, just because of the type of sports males participate in. Okay. And so now that we've mapped a specific region on the VEGF gene, we could look at the functional significance of those sequences in sequence to see how that potentially affects messenger RNA and protein. And then hypothesize, remember in the earlier I showed you the dogma, it goes from DNA to RNA to protein, cell, tissue, disease. And so we want to go back to how can we translate what we see at a statistical level to function and clinical phenotype. Similarly, we did a KDR haplotype, and we found a specific disease susceptibility haplotype in males and females. When we looked at Achilles tendinopathy, we did different populations, because it's important to you want to look at different populations to see is it biologically significant. You also want to look at different haplotypes to assist you in walking along the chromosome to see which way must you go, more left or more right, because you're still trying to explore the potential functional susceptibility locus. And so we've explored Achilles tendinopathy in a British population as well as a South African population, and we found the AGG haplotype in the vascular interferal VEGF-A to be associated with an increased risk, and we didn't find any association when we looked at the UK population. But when we combined the populations, and we only combined the populations when the frequencies of these individual polymorphisms were very similar, and so we saw that the AGG frequency is still associated with an increased susceptibility in both these Caucasian populations. And so we've looked at several populations, and I'll talk you through this slide. So we've looked at a chronic phenotype, which is our Achilles tendinopathy. We've also looked at acute phenotypes. We looked at 2 ACL populations within South Africa, and we've also looked at acute Achilles tendinopathy rupture. So we wanted to see, is there a different VEGF or angiogenesis profile showing a risk if you have a chronic disease, a chronic injury, or an acute injury. And so the red reflects increased susceptibility, green decreased susceptibility. And if you look down here, we seem to find that with ACL ruptures, there seems to be a GA increased susceptibility, which we also see with acute, but we don't observe it with tendinopathy. We also see with the VGF A haplotype, we have an increased risk with females, as well the alternate haplotype is associated with an increased risk of tendinopathy. And if my memory serves me right, that the AGG is associated with an increase of, a decrease in angiogenesis. So they have less blood vessel formation with that particular haplotype. So we've shown that specific functional VGF inferred haplotypes are associated with ACR ruptures, and that the alternate haplotypes are associated with susceptibility in tendinopathy. And so even though tendons and ligaments are structurally similar, they function very differently. And so it may be that an increased angiogenesis is a protective mechanism in one connective tissue, but it is a risk factor in another connective tissue. And so when you are prescribing exercise intervention or therapy, one has to therefore be mindful at what therapy is going to promote new blood vessel promotion, thereby increase your healing response, or when it's not appropriate and where you will be increasing your susceptibility to a re-rupture or a chronic injury. And so one always has to function, even though you're doing something statistically, you need to go back to biological mechanisms to try and explain what is happening within the connective tissue and what is the clinical relevance. But the angiogenesis pathway doesn't function by itself, it functions within the body. And so there's a lot of other signaling happening. And so if you look at your several studies have shown that mechanical load upregulates our different cytokines, such as this, your interleukin 6. And interleukin 6 also have a tendency to upregulate your angiogenesis pathway. And so I show new polymorphisms within the angiogenesis pathway, but we have to be mindful of what is the genetic contribution to your genotype score in your other pathways. And so we, in a very small sample size, we know that is a limitation. And that's why we're here, so we can increase our sample size and work together in creating a more correct model. So when we looked at all the associations within the interleukins, the angiogenesis pathways, we actually see in the tendinopathy and risk model that the VGF, age and BMI are still the biggest predictors of increased risk compared to our interleukins. So what do we know? We know that there are specific genomic intervals that we can now study at a functional level to be associated with either increased susceptibility or decreased susceptibility. We've narrowed down, it's not just X amount of markers, but we've narrowed down a starting point within those genes to look at functionality. And so you need to do the next steps to look at function. You need to repeat it in different populations. You need to understand potentially what is the effect on clinical treatment or clinical intervention. And we spoke about that neat parallel fibril structure within tendons and ligaments. And so we want to know what are the components that play a role in regulating that. Some of them, besides type 5 collagen, a lot of work George McConey from Botswana did on type 5 collagen. He was the initial PhD student in the group. But there are other genes also involved with regulating fibrinogenesis. These are your proteoglycan genes. And so we wanted to identify, are there markers with sequence variants within these proteoglycan encoding genes which may contribute to susceptibility to connective tissue disease. And we've looked at different disease pathologies, Achilles tendinopathy, and in South Africa as well as in the Polish population. And what did we find? We found that even though these are very small samples, and we've implicated specific variants within the aggrecan gene to be associated with a reduced the white bars being your controls with a reduced susceptibility to Achilles tendinopathy. We've implicated the same polymorphism, but with the alternate that the TT is now associated with an increased risk of Achilles tendinopathy in a British cohort. And so ideally you wanted to see the same relationship with the increased genotype related with an increased phenotype in both populations. So we have to think about, are we away, are we within the region of functionality within that gene, or are we just slightly away? So we're trying to map that particular region to see which way to go. And we also looked at it in a, when we looked at the ACL injury, we also found similar. We didn't repeat those findings, but it's still within that same gene we found an association. And so we need to look at more polymorphisms and do a haplotype analysis around that region. Similarly with Decadin, we found that in the South African population we found a GG to be associated with decreased risk, and in the Polish population we found that the AA is associated with an increased risk. And so in different populations it seems at the proteoglycan level we're finding alternate genotypes to be associated with risk or protection. And so either one can say that those associations were not real, or we are distance away from that genetic interval in that specific gene, so we need to explore it more in detail. But I was interested in, those are my pet genes, the proteoglycans, and so as part of Kyle Willard's PhD thesis, we decided to explore these polymorphisms functionally, and we did biopsies of skin tissue with just here in the crease. A clinician took a small biopsy and we constructed fibroblasts from these tissues, skin fibroblasts, and we genotyped them. So we had different cell lines where we knew the exact genotype profile, where those who were associated with the decreased risk for the biglycan locus and the COL5A1 locus, and those associated with the genotype associated with an increased risk. And then we looked at the COL5A1 messenger RNA expression, and we found that there was differences in the messenger RNA expression between the different disease risk genotypes. So with the decreased risk group, we found more COL5A1 messenger RNA being produced. Compared to the increased risk group, we found less COL5A1 messenger RNA being produced. We also treated these cells with transforming growth factor beta. Why did we choose that? TGF beta is another Nobel Prize waiting out there. It is the switch between the healing and fibrosis. And so we want to understand what is those, and TGF beta also regulates proteoglycan synthesis, and proteoglycans also regulate TGF beta synthesis. So there's a real negative feedback loop within the tissue of, so if you fine-tuning within the tissue's response to load. And so we wanted to make sure that proteoglycans are going to be produced. And so we added some TGF beta, so that, and heaven below we saw, if we treated the cells with TGF beta, we saw an increase in these proteoglycans, in these COL5A1 messenger RNA being produced, and we saw similar trends, similar statistical association with the decreased genotype profile being associated with an increased COL5A1 compared to the increased susceptibility. Similarly, we looked at biglycan messenger RNA expression, and we saw more biglycan messenger RNA expression being noted with a decreased risk at the genetic level compared to the increased risk. And when we treated with TGF beta, we didn't see statistical difference in our risk groups, but we did see a fourfold increase in the amount of biglycan being produced. And when we looked at the Decadin messenger RNA expression, we also saw a difference in our messenger RNA being associated with a disease profile, a disease risk profile. And so we're going from association at a DNA sequence level, where we associated with the clinical phenotype, and we're now showing at some functional level, at the messenger RNA level, that there is some change in messenger RNA being coinciding with the risk at the genetic level. Similarly with the decadent. Okay, and similarly we found with the VGF messenger RNA expression, VGF being your angiogenesis pathway, where new blood vessel formation is increased, and this is clinically relevant, you understand. Is this VGF risk haplotype or genetic profile associated with more VGF being produced, and is that increase your risk susceptibility? So if you prescribe a specific treatment which is contradictory to the person's genetic profile, you need to understand the individual connective tissue and the prescription that you're giving. And so we looked at VGF messenger RNA expression, and we found that a decreased risk is associated with an increased VGF messenger RNA compared to your increased risk. And similarly, when we treated with TGF-beta, we found the same statistical significant difference and a fold increase in VGF messenger RNA expression. So in summary, we're seeing a statistical trend with the level of messenger RNA being expressed with a particular disease phenotype. So going back to my earlier statement that the genome is so large, and yet we're only implicating a very small number of candidate genes. Those are the genes that we know of, that we've sat down one day and said, oh, these are my pet genes, they function such in the connective tissue, let's have a look at them. So there's no, so it's just by chance that we found these associations. And so we wanted to develop a multi-step approach to identifying this missing heritability. And with technology being available, we went with a whole exome sequencing approach. We sequenced, and this paper has now been submitted to +1, and it's in its second phase of review, and all our sequencing data is available on their website as well if it gets accepted. But we went through whole exome sequencing. It's quite expensive. And so we couldn't, like we do with the genetic association studies where we look at about 600 samples, we didn't have money for that type of analysis. And we thought of what kind of strategy can we come up with, and we went for a strategy where we looked at exemplar cases and controls. We took 10 controls who were much older than our cases. They had no history or family history of either tendon or ligament injuries, and they were our controls, and so they were also represented as a extreme phenotype. And we sequenced 10 cases. They were young age of onset, less than 35. They had multiple injuries, and they've also had bilateral tendinopathy. And so those were our extreme cases, and we sequenced them, and we wanted to see, yes, it's a small sample size, but it's a pilot study, right? And we wanted to see, can we pick up variations which was more, and so these polymorphisms that we look at are common in the general population, right? And so we just thought, can we pick up polymorphisms that are more frequent in these extreme cases or more frequent in these extreme controls? So the controls will have a reduced susceptibility, and our cases will have an increased susceptibility. And so that was the approach we followed. When we did the whole exome sequencing analysis, the bioinformatics has flagged about more than 200,000 variants to be potentially different between the cases and controls. And that would take us a month of Sundays or a year of Sundays to analyze, let alone the financial status, and my PhD student would not accept the study. So we thought of a more creative way of looking at this huge amount of data generated from these 20 samples. And we said, well, let's think about if there's a 30% differences between the frequencies of these polymorphisms between our cases and control, what proportion of those polymorphisms can we extract? And we did that, and we went from 200,000 to about just under 4,500. Then we said, that's still a lot of sequence to look, because we would then look at those in our our cohort of about 600 samples. And so we said, well, let's look at the previous candidate genes that were implicated, those 80 loci that were implicated, and how many of them are these variants that we picked up, this 4.5, are residing within these 45, in these loci. And we've also developed a bioinformatics tool, which, and I'll explain that to you later. And so we also use this bioinformatics tool to extrapolate potential candidate genes that have been flagged by this whole exome sequencing analysis. And then we used various in silico tools on the public domains to see what are potential functional. Remember, we were looking at those different genes, and you should, and there are tools to show you what are the functional regions within those genes. And so we also looked at, okay, extracting those polymorphisms, but we actually want to know polymorphisms which have some, it's proof of concept that they have some function. And so we looked at those as a target polymorphisms to look at. So this was a very intriguing filtering strategy that we had used. And so that is why we submitted it to +1 as a strategy for others to use in understanding complex phenotypes. And so this in silico tool that was designed by collaborators at the University of the Western Cape, they called it the Borg because he was a Star Wars fan, and the Borg stands for biological relationship graph. So it incorporates all the information on the public domains relating to the different animal models to human models. And the PhD student, Senelele Dlamini, she looked at the clinical phenotypes and descriptors for Achilles tendinopathy and ACL ruptures, found out what is the root terms used within these various databases, plugged that into a machine learning tool so that we can just plug our whole exome sequencing data into this machine, or we can plug in the whole genome sequence into that and come up with potential candidate genes based on data that's already out there in the public domain. And so we did that as well. And so using this filtering strategy, we went from 200,000. This first bar is the chromosomes. The number of chromosomes there are is 1 to 22 in X. And this is the amount of polymorphisms on chromosome 1 alone, not just under 20,000 polymorphisms. Then we looked at the previous candidate genes that we've implicated in those 80 loci, and we found the 30% difference between our cases of control. And so we moved down from a considerable amount of 19,000 potential polymorphisms to investigate to 1 polymorphism in this particular gene, 6 polymorphisms in that particular gene. So this was a filtering strategy for us to see what are the specific polymorphisms, potential polymorphisms, which are biologically significant to investigate within our cohorts. This is just another diagram where these are the previous interesting candidates that we've looked at. The PhD student, she submitted, she's came back with glowing reviews, and I'm the supervisor and I just check her corrections, which I haven't done yet. So I'm the delay in her process of graduating. So, don't tell anyone. So this is the particular gene that we were interested in, called 27 and tenascin C. Why were we interested in those two genes? As part of my postdoctoral research years ago, we've looked, and Colin Sanders, the PhD student, we looked at polymorphisms. So COL27A1 and tenascin C are lying next to each other. So we looked at polymorphisms and we implicated a haplotype. Remember, it's when you're looking at a block of polymorphisms which are inherited as a chromosome block, and they spanned both these genes. And we weren't sure, is the disease susceptibility localized to COL27 or was it localized to tenascin C? And so we wanted to tease this out with whole exome sequencing, and so we managed to identify new variants for us to explore to narrow down a potential biological path in these genes. And that's what she'd done. So what is tenascin C? Tenascin C is a hexameric glycoprotein, and it plays a role in the healing response of tissues to load, and allows for, you can see from these arms, it allows for communication between the fibrils. And so we've mapped all these polymorphisms to the tenascin C gene, and by doing genetic association studies, we've identified specific polymorphisms, specific genotypes in this polymorphism to be associated with an increased risk in tendinopathy, and another polymorphism to be associated with increased susceptibility in ACL ruptures. And so using haplotype analysis and linkage disequilibrium, which are just genetic tools, we were able to localize a region towards the tenascin C gene to be potentially be involved in susceptibility to ligament and tendinopathy injuries. These are the haplotype analysis that we found. And then, again, we can't just, genetic association study is really a statistical association, right? And so you want to look at what is the functional significance of this work. So we used some in silico analysis, and we found that there are some suggestions that there is some functional role of these specific sites within these genes. And as part of Andrea's work, we're quite fortunate that we've established collaborations with different partner institutions where our graduate students can go and spend 6 months there, and their students can spend 6 months in our labs. And she was able to go to Manchester University to look at some functionality. So what she did is she looked at specific haplotypes, and then the alternate, so it's an increased susceptibility and the alternate haplotype which is associated with a decreased susceptibility, and she looked at functional organization. And so there's some preliminary evidence from that, which I'm not going to report on today, to show that there's some functionality of that disease susceptibility locus. And so hopefully I showed you how we're trying to use next generation sequencing technology to map functional regions within our genome and then show functionality that we could start exploring those regions for susceptibility. And so before we can predict susceptibility and predict risk management and tailoring precision medicine for an individual, we really need to understand what's happening at the molecular level. Is it real what we're seeing? How true is it at the functional level? And what is the clinical relevance in different populations? Because we're not only treating people in South Africa, but we want to treat people globally and understand how people respond to load, what is driving the capacity of a tendon to respond to load. And even though I showed you examples here with 2 common sports injuries. However, if you think about connective tissues all over the body, we're also busy with a project with a collaborator of ours who's looking at breast cancer treatment and the disability depending on what treatment you're receiving. And we find that those who have an increased disability or pain score actually have some of the risk loci for these tendon injuries. So the same risk-associated genotypes with Achilles tendinopathy and ACL ruptures are also associated with a more severe disability score for these ladies who've undergone specific breast cancer treatment. And so you need to understand how does a genetic susceptibility of a connective tissue respond to different load and interventions. And so even though there are many direct-to-consumer testing out there, it's still premature. We actually do not understand what is happening at the genomics level, because as I said before, that these polymorphisms are within all of us. So we need to understand the body as a whole and not just at the sequence level. And future work, and good future work, is dependent on collaboration, and good collaboration between all types of biologists, clinicians, so that we can correct what is wrong, increase what we don't know, and understand how everything works together. And I would like to acknowledge the team back home. These are some of our members. Our head of department is here, Professor Malcolm Collins, and he started all this work many, many moons ago. And I joined the research team in 2005 as a postdoc. And so we are now co-PIs on the team. And we've received our funding bodies, our collaborators in the different institutions, and I thank you for your attention, and please come and visit us. We have sharks, and we've got, what do you call these things? Penguins. And thank you for your attention.
Thank you very much, Alison. I think one word I caught there is the knowledge barometer. I think it's very interesting. So at this point, I think we will invite people, questions, comments, and so we probably take 3 questions at a time and then move on.
Thank you for. I can, yeah, it's fine.
Yeah.
Really interesting. I really appreciate you want to talk about this. One of the things I really enjoyed was your SNPs for ACL injuries with contact and non-contact sports. And kind of the big thing in genetics now is DNA methylation epigenetics.
Yes.
I'm just curious your comments or thoughts on how that might play a role in the contact sports. Is there something that is going to lead to DNA methylation that would influence injury risk or not?
So I think DNA methylation doesn't decide on contact, or doesn't decide on the mechanism of injury. DNA methylation is the is the body's biological tool to switch genes on and to switch genes off. So depending on the environment that it's functioning in, it will switch it on or switch it off. So that is a very important step for us to look at. And a lot of the microRNA, which is small RNAs, is an important role that we need to start looking at as well. And so people are using that in different diseases to prognostically say who's going to be responding to a different treatment. And so I think in connective tissue, we really need to start to understand epistasis, and that is a disease mechanism. And so we wanted to look at microRNA regulation, but with microRNA, you need to understand when to take the tissue, because when it's going to be switched off and when it's going to switch on, and then how you're going to compare it. So there's a lot of understanding still that we need to do before we can dive into experimental design.
Hi, thanks for your talk. I was just wondering if there are any groups out there who've developed any mouse models that are targeting these specific genes, and then take those into kind of like a injury biomechanics aspect.
So there are mouse models for tendinopathy, and they've looked at a lot of work on the collagen type 5 gene and collagens. So we're still premature with the other pathways, and I think now that they've shown with vascular endothelial growth factor and angiogenesis, there may be more interest in looking at the angiogenesis pathway, because before physiotherapists said, well, you know, that these tissues are limited in their blood vessel supply, so why would that be a disease mechanism? But now when they're showing that actually that could be a protective mechanism as well. So I think the next wave is going to look at these. So it's a collective idea, and people sharing and communicating and understanding. Those are new ideas to explore at a functional level.
Really, I'll be all right. So very interesting work. So I come back to, like, in tendinopathy, loading parameters are very interesting. Like, if you're more susceptible or less susceptible to load, that may help with regeneration of the tissue, or at least keep, you know, some healing or angiogenesis of the tissue that's on. In the ACL, if we're talking about like an acute injury mechanism, so identifying susceptibility is important, right? So there was, I mean, for 20 years we tried to identify that women were more susceptible than
Yes.
men, right? I don't know why it took that
And we
long,
still
but
don't
it
know
did,
why.
right? And now we just keep combining, you know, again and again. But I wonder if we were able to identify a genetic marker that says you're more susceptible for acute non-contact ACL, like, does that just tell us that we should do a neuromuscular training program with those individuals, or is there a more epigenetic or genetic intervention that would sort of be the next step to help change tissue properties?
So I think we can't have a one system fits all. We need to understand how they all work together and how the tissue functions within a different environment. So I don't think it's one or the other. But you want to understand, so even for females, why females, is it just hormonal, or is it the structural component? And often the structure and the biomechanics is also genetically determined. So there's many functions of your ankle and your ligament which are genetically determined. And then the load is just another parameter on top of that. So it may be that these associations that we're finding is related to structure and function, and not necessarily how we're responding to load. Correct? Yeah.
Any more questions? Al?
Okay. Well, please join me in thanking Dr. September. token of appreciation.